background than the patients, and it requires immunosuppression. We are still searching for exactly what is the minimum immunosuppression that's required and for how long. Right now, all patients are required to take all immunosuppression medications as long as they can tolerate. Some of the patients, about 1/3 of the patients, could not tolerate it beyond a few months and then dropped the medication. Others were able to tolerate and continuing. And we don't really see much difference in between the two populations, but it's really too small, too early to make such conclusions. But this is the list of actual medications that are on the protocol. So there are 5 treatment groups that are in the trial, and these treatment groups are designed to test the safety versus benefit. So because it's a first-in-man, in discussions with the FDA obviously stressed the safety aspect of it more than in other words, more on the risk than on the potential benefit. So that the first group of the patients we treated were non-ambulatory patients, meaning that they lost sufficient amount of the motor neurons in the lumbar spinal cord that we most likely they didn't have any that we could protect. But nevertheless, that allowed us to test potential risks involved in the surgical procedures. The next stage were ambulatory subjects, and we started with the low doses, just injecting the cells on one side of the lumbar cord, and then in the subsequent groups, we increased that dose to both sides of the spinal cord, and then in the next cohort, we injected the cells into the cervical segments, which are conceivably a little more risky should anything go wrong. And then in the final group, we are injecting both into the cervical segments as well as into the lumbar segments. And right now, presently, we have completed Group D, all 15 patients, and we're about to start on Group E patient, which you are able to enroll the previous patients that were previously treated in the lumbar segments now to receive a cervical injections as well. The side effects basically we saw was mostly related to the immunosuppression protocols. There was really no cell or surgery related complications. The data that I'm showing you only of the last 6 patients, 7 to 12 patients, and in summary, basically the FVC, most of the patients have stabilized FVC. ALS, FRS, some patients have improved, some have stabilized, some continue to decline in similar patterns in other measurements of motor function. And the next phase we're still discussing, but we have proposed to the FDA to enroll additional groups into this study in order to test escalation of cell doses. Thank you for your attention.
For giving a really detailed explanation. As you recognize, it's a very complicated procedure, very unlike taking medication once or twice a day. And so the caution and the rigor that's gone into the study is quite commendable. So our last speaker before we open up to a discussion is Dr. Kerr, who will give an update of the Biogen study, and it's a pleasure to have him present it. He actually has, in the past, worked a lot in SMA and was at Johns Hopkins, and we feel thrilled that such a talented and committed clinician is really leading the team at Biogen.
Thanks for having me. Thanks to all of you for your efforts locally and at the national level with the ALS Association. I'm glad to do this. This is, I think, probably my 5th time to do this, but it's only my 2nd time as a member of Biogen Idec. Prior to that, I was at Johns Hopkins. I'm a neurologist. I'm an ALS neurologist. I have done a lot of work on motor neurons and spinal cord, but I'm now directing our efforts in neurodegeneration in Cambridge, Massachusetts at Biogen. And I'm going to only actually show you about 4 or 5 slides. I know you've had a very busy day, a lot of slides that go by very quickly, so you're only going to get a few from me, and then we can just do questions. Maybe that's the best way. I'll tell you very briefly about Biogen. Biogen Idec has been around in Cambridge, Massachusetts, right between Harvard and MIT for the last 35 years, and has been largely a multiple sclerosis company. That is, we have developed drugs for MS. And what we've done over the last 4 years is to shift, in addition to multiple sclerosis, to looking at neurodegenerative disorders. Neurodegenerative disorders are the class of disorders like ALS, like Parkinson's disease, like Alzheimer's disease. And we think that we can dive very deeply into understanding what these disorders are, how they are similar in some respects, and potentially develop therapies for them. So our lead effort in a neurodegenerative disorder is with a drug that I'll just call DEX. Dexpramipexole is its full name, but it is a drug that we are testing in a phase 3, very large ALS study now that will read out whether it works or not around Christmas of this year. I don't have any data to share with you. I don't know whether the drug is working. I have no idea at all because it's blinded and randomized, as Dr. Berry has told you. But I will tell you how we're thinking about things in ALS. So Dr. Berry shared this with you, and I won't go into this into too much detail, except to say that top box is really that there is a trigger in ALS, and it is probably different in different people. In some cases, you've heard about genes and genetic mutations. In some cases, it may have been an environmental exposure. We don't really know what the triggers are, but they're probably not the same for every single person. They're different. But the middle box is kind of what probably happens to most or all ALS patients, regardless of what their trigger was. A series of things happen to the cells that are in the spinal cord and in those nerves, those special nerves that help you to move. So mitochondrial dysfunction is one of them. That's one I want you to remember, because that's how we think dexpramipexole, or DEX, may be working. Mitochondria are the powerhouses of your cells. They are the things within your cells that generate energy and allow your cells to work, especially neurons, but it's true of all cells, that they require a high amount of energy, and the mitochondria are critical for this. Dr. Berry also talked about these other things. So metabolic dysfunction. It turns out we use energy substrates differently in ALS compared to normal people. Proteins aggregate together. They form these large intracellular aggregates that kind of mess up how the cell and the neuron works. You heard about glutamate and growth factors, and these are the cells that are around the motor neurons themselves also start to work badly, and then the cascade accelerates. Ultimately, what you're left with is synapses that aren't working. These are the connections between neurons. The axons are the long cables or wires, and ultimately the neurons die. And that is why people with ALS get weak. So Biogen is very interested in this. We think an awful lot about these mechanisms and how we can potentially target them and attack them to stop neurodegenerative processes. So there are several buckets that go back to that middle box that I was talking about in the last slide. Metabolic integrity, one example of that is mitochondrial dysfunction. If the organelles, the mitochondria that produce energy for your cells are not doing so correctly, you're going to have trouble. And so are there any therapies that may increase metabolic integrity, increase the efficiency of mitochondria? And there may be. We're at the very beginning of what I hope will be a combination approach to ALS and neurodegenerative disorders. The middle box there is structural integrity. So ultimately, we have to figure out ways to protect the axons and the synapses and the neurons from dying, or maybe getting them to grow back. And maybe Dr. Joe talked to you about the stem cells. Maybe they can help keep the structure of the spinal cord intact while we're fixing the things that are going wrong. Oxidative integrity. So you've heard about oxidative stress, and that certainly happens in ALS, and we're just starting to see some therapies that may alter oxidative stress in neurodegenerative disorders, and ultimately macromolecular integrity. That's this misfolding or aggregates. We think there may be some strategies to make those aggregates melt away so that the cells can work better. So this is how we're thinking about ALS, is as you work from left to right here around this wheel, we think that there are several things that we need to do better to develop better therapies for ALS. First, it starts with early and accurate diagnosis, and we need to diagnose ALS earlier than we currently do. In some cases, it may take 12 months, 15 months, 18 months to actually get the diagnosis, and we'd love to be able to make that diagnosis earlier so you can start therapies earlier. But we need new therapeutic targets, so we need the basic scientists to really dig in and figure out what is that research, where are the things that we should go, and we are doing that as well as many other groups are trying to identify what are those areas where we can intervene to stop this process that causes these neurons to die in ALS. And then that long process from a target, meaning something we should block, to a drug. And Dr. Berry talked you through that long line of investigation that is required to go between those two steps. But ultimately, I do think we will need combination therapies. I think one therapy isn't going to work. I think we need to try them on multiple approaches in parallel. And you can think back to even 5 or 10 years ago with, for example, HIV, which was relentlessly progressive and universally fatal, until combination therapies were employed that then could halt HIV, and now patients with HIV can live for longer periods of time. That's the kind of approach that we need to develop in ALS, not just companies, not just nonprofit groups, not just academics, but all of us working together to figure out what are those combinations. Are they safe? Can they work together? Can they add together to increase the treatment for ALS patients? Pharmacodynamic biomarkers is just a fancy term that means we need to know early if a drug is safe and is working. We can't wait for 7 or 8 years and 3 stages of clinical trials and thousands of patients. This is too serious a disease. We need to know that early so that if a drug doesn't work or if a drug isn't safe, then get rid of it and let's move on. Let's move on to another one that has a shot and keep testing it. So all of these things are part of the landscape that we now need to change, and we're working very hard to do that. So Empower is the name of the phase 3 study that I told you you will hear about around Christmas at the end of this year. Empower was first studied by a company called Knopf Biosciences out of Pittsburgh, Pennsylvania, who did a very nice phase 2 study. That was the second study. It was the first study in ALS patients. And in that study, there was no significant benefit, significance meaning was statistical significance. The drug looked like it potentially worked, but we don't know for sure. The drug looked like it was well tolerated, but it was a small study. The one thing that we knew from that phase 2 was not that it worked, not that it was safe, but that it needed to be tested. That we needed to do a very rigorous test as quickly as possible to see if it had effectiveness and is safe. And that is how we designed this study called Empower. And this study was designed to replicate the study population from phase 2. In other words, we had an encouraging finding in the phase 2 study. The phase 3 study had to replicate those findings. So many of you have asked me, why couldn't you take patients who have had ALS for a longer period of time? The goal here is to get this drug approved if it is safe and effective. And the first step is to replicate what was initially studied in the phase 2. And then we can go out from there. And so we designed the inclusion and exclusion criteria identically to that of the phase 2 study. And the study drug and dose and dosing regimen were identical, which is 1 tablet twice a day. And the goal of the phase 3 study is to not only know if it works, but also to know if it is safe in a large number of patients. So what we did is we identified the target number of patients, and that was 804 patients. But we actually had such an amazing outcry for participation in this study that we screened over 1,000 patients, actually 1,118 patients. More patients qualified for the study than we had anticipated, and therefore we ultimately kept in the study not 804, but 943. So one of the largest studies in ALS that was ever done. That included 83 clinical sites with investigators at each of those sites, of which 80 of those sites actually enrolled patients into the study. And if you look at this graph on the left, you can look at, say, the blue line. This is the screened patients by month. And you can see that basically all of these patients were enrolled in 5 months. And so this was not only one of the largest ALS trials ever done, but it was the fastest recruiting ALS trial ever done. And again, that's a testament to not that we know the drug works, we don't, but we need to test it as quickly and fully as possible so that if it does work, and if it is safe, it's got to be made available to the ALS community. And we're very passionate about that and very committed to answering that question if this is a safe and effective drug. Most of the patients, or the largest group of patients, came from the US. This study was also done in many countries in Europe, as well as Canada and Australia. So this is my last slide then. So the phase 2 study was encouraging, but it was small. There was a suggestion of a benefit. It could be real, it could not be real. We don't know. But we felt like we needed to study this rigorously in a phase 3 studies, which we've done, modeled after the phase 2. The primary outcome is a functional outcome, which you know about, called the ALS-FRS, Functional Rating Scale, as well as survival. And the next steps are, there are other studies called clinical pharmacology studies ongoing. In other words, it's not just that phase 3 study called Empower. There are 7 other clinical trials currently underway with DEX to see the safety of this drug in various populations. We need to know if it does work. We need to be able to tell you what drugs could you take with it? What drugs shouldn't you take with it? Can you take it with food? Not food. How about if you have some renal problems? These are the types of questions that we need to answer, because if we don't answer them and we just provide it to the community, it could cause problems, safety problems, adverse events. So all of those will be done in time for our Christmas readout this year. We don't know if that will support approval of DEX for ALS. We don't know if it will work, and if it does work, we don't know how effective it will be. What we anticipate is that we will look at the data very carefully, and if we can, file to the regulatory authorities like the FDA in the United States to get it approved for ALS. We certainly will try. So I will stop there, and then we can take questions as a panel. Thanks very much.
Staying very nicely on time. We have about 25 minutes for questions, so really anything related specifically to what you've heard today, any questions you have in general about clinical trials, this really is your opportunity with all our experts here to have the opportunity to ask the questions. And Marsha is observed.
First is for Dr. Berry, I think it was the second speaker. He talked about placebo. This is just my opinion as an ALS patient and pal. ALS's pals that I talk to actually have the reverse placebo effect. Everybody is afraid of getting a placebo, where a person with a migraine thinks it's going to help. How not giving them placebos and just trying the drug, because everybody I talked to was willing to take a chance because they don't have a year to blow away on a placebo.
Thank you for the question. I think that's your question is definitely exactly on target. I think if we could design a trial to, I'm sorry, I'm going to just, okay, let's stand over here. I feel I want to be able to look at you. So I think if we could design a trial that didn't have placebo and was able to show, to demonstrate conclusively that a study drug was active, was effective in treating the disease, we would do that. There are, and I think we make big attempts to reduce what we think of as the placebo burden in a study. That is to say, we could design a study where everybody from one patient in the study to everybody in the study was getting active study drug. And we've already said that we don't think we can probably design a trial that proves beyond a reasonable doubt that the drug is working if we had everybody on the active study drug. But we can try to reduce the percentage of people who are on study drug by changing the percentages. So we might say 2/3 of people will get the active study drug and 1/3 might get placebo. We do that in an attempt to reduce this placebo burden. But I'll tell you that once we get the statisticians involved, then things get more complicated. When we do that, when we reduce the number of people or percentage of people who are getting placebo, we give up some of what we call statistical power. We need to enroll overall more people in the trial. So there's sort of a balance in being able to answer our question with the fewest number of people and as quickly as possible versus getting as many people to study drug as we can. So we're constantly trying to come up with ways to overcome this. Another way is to have an open label part of the study, an extension of the study where everybody gets study drug. But this is something that I think is a huge issue in designing these trials. Thank you for your question.
All right. One more question. This is for Dr. Joe Ford, if I'm pronouncing it right. I received a stem cell infusion of mesenteric, if I'm pronouncing that right, also stem cells that were from a donor. And I took no suppression drugs at all and had an infusion in my lower lumbar. And it worked for me, and it worked for the 2 other people that I was involved with that know personally. And that's all I can say about that. And I think stem cells is the way to go.
I wanted to ask a question in regards to barriers in research. I always ask this to every panel. You all have some individual thoughts on particular barriers in research. Is there particular barriers that you see in this disease, ALS, and can we help? We want to help. The ALS population wants to help. What can we do to help?
Yeah, so I can start there. There are probably a couple that jump right up at me. One is the models that we use to get drugs into patients. The SOD1 animal has been very valuable to us in understanding the disease, but we need better models. And some of these models could even come from stem cells that are not used for transplantation, but are used to screen whether drugs can be effective in ALS. That would be a lot quicker than going through animal models. And the animal models haven't been particularly predictive. And so better models for us to test new drugs that would more efficiently allow us to get into patients would be certainly one bottleneck. And the other one I mentioned in my talk is just that we need an earlier read on whether a drug is having any effect in an ALS patient. And you know that ALS patients can be very different in how they progress, the rate at which they progress, the symptoms that they have. And so it's sometimes very hard to tell if a particular therapy is doing anything. But so far, what we need are literally hundreds to thousands of patients for months to, in some cases, years. And if we could do this more quickly, and there are some biomarkers that are being developed that may do this, we would be able to more efficiently go into ALS trials. And when I mean biomarkers, maybe it's something in the blood, maybe it's something in the spinal fluid that tells us you're either doing what you want to do or you're not. Or maybe it's a muscle measurement, or maybe it's an electrophysiologic measurement. Something, though, that gives us a sense earlier with fewer patients so that if it's not working, we can move on. If it is working, we can pick the right dose and go to the larger study.
Yeah, no, I guess I would mimic that comment. I think biomarkers in the disease are an active area of research that if they could act as outcome measures for clinical trials, or ways to diagnose people earlier, or ways to direct people to the appropriate clinical trial for them, I think this would be a huge advancement in our program for clinical trials.
I'd like to comment actually on what that gentleman up front just said about the issue of using the placebo versus the drug. Now, a lot of people can't get to the studies. Okay, there's not enough ALS folks. That was one of the problems with getting to some of the studies, and not everybody can afford to get to the studies. So actually, I would hazard to say and possibly think a little bit outside the box, and a lot of people have MRN nurses and things that can go and do the FSR checks and things like that, as opposed to those that can afford to go to the studies. So you actually do have a control group. Just maybe think about using the control group as those folks who can't get to the study. Just a thought. My other question was kind of directed towards the DEX study. Coming up in December, hopefully some results will come out of that. It's going to go to the FDA. I understand that ALS has received the ability in clinical trials to be moved forward quickly with lesser numbers in each of the category, in each of the phase 1, phase 2, and phase 3. What about an FDA fast track? Has ALS got an FDA fast track? Is there, what's the process and about what's the timeline?
So that's a very good question. So Dex pramipexole for ALS has been given a fast track status, which means that the FDA and in Europe, the European Regulatory Authority will evaluate this as quickly as possible. I must tell you, the FDA has met with us many, many times to help us think about the design. They have really been partners as we have developed this, not adversaries. They are very committed to bringing therapies forward. They are just very committed to making sure that it doesn't harm people. So they will do it as quickly as they can. They are committed to doing this within an 8-month time frame to review the packet. Now just remember, the filing packet that goes into the FDA is on the order of 30,000 pages. And it's a lot. Every single person who was in the study needs to be detailed, and all of the adverse events, so that the FDA can make a complete picture. And again, the reason they're doing this is because they want to make sure that this drug is effective for patients and isn't causing some patients to actually have an accelerated or worsened disease. So the expectation would be, again, it depends on whether the drug is effective, but if it's really effective, we will submit it potentially in 2013, and it may take up to a year actually before it gets fully approved. Probably a little bit less than that, but we'll see.
Yes, this question is for Dr. Berry. You might want to stand up. I was part of a clinical study this past year, and it's been a roller coaster here. I went from not very good to about midway through the study doing really great. And the study's over, and I'm declining. So, I came here kind of for a compassionate plea for the medication. I know you hear this a lot. And this particular medication was in clinical phase 2 in the efficacy study, complete by the end of calendar year 12. So my question is how do we continue to get medication that is working before FDA approval? Or, and I'm not sure I need to mention the name of the drug, but the gentleman from Dex is here, or the power study in Endeavor, I guess. So it was in P-01. So that's my question.
So it's a good question. There are, I'm going to have to take a step back and sort of explain the processes to you. And the process is typically when a clinical trial is designed, there's a decision about whether the drug can be offered on what we call an open label extension at the end of the trial. In other words, for some trials, patients who participate in the trial can then be offered, whether they were randomized to placebo or to active study drug, can then be offered the study drug on an ongoing basis. This is often done with phase 3 trials. It's sometimes done with phase 2 trials, and there are a couple of considerations that dictate the response. One is how much data there is about safety, dosing, and whether it's allowable. So it may be in phase 2 that we have enough experience with the drug that all the regulatory bodies feel comfortable saying, yes, you can have an extended time period that people who are in the trial then have open label access to the drug. But it may be that there are still questions about the safety that need to be answered before allowing open access. And the second one is production and financing of that. So it's a complicated process to continue to make drug at large volumes. So
people that, actually one gentleman was an MIT professor, claims he's gotten a patent on this drug, and he's on patients like me, and there's 28 people making their own medication. And I think that's not what we want to happen. Somebody's gonna, injure themselves, it would hurt our cause.
Yeah, no, this is a tough one. I'm not sure I can answer it, but I agree that having people make their own drug is not necessarily better.
I mean, if I could just,
the first choice.
if I can just add to that, I know there's a huge unmet need, and people would be tempted to synthesize a drug and take it outside the context of a clinical trial. And the downsides of that are, we really don't know if that drug, or any drug, until it has been approved, is safe. And in ALS, we've been there before, where we thought a drug was effective, and when it was tested further, it was found to not only not be effective, but actually in some cases worsen the disease. So there are several examples of that. And so we have to be very careful, because if a patient were to take a drug outside the context of a clinical trial and had an adverse event or a death or some consequence, it would put the whole clinical program potentially on hold until the FDA evaluated this, and it would delay a drug getting approved. So there are risks to that, and if it is synthesized outside the context of the clinical trial, you don't know what you're getting. It may or may not be what you think you're getting.
I'd just like to make a comment about CK357. I participated in the A portion of the phrase. It was very effective. Left-hand strength went up almost 50%. Right hand almost 30%. But last week, I asked at the clinic if they had a black market supply. The other thing is that over a year ago, on the Cretan study, it's still languishing. It hasn't been filled. That's over a year ago. So you wonder why it takes so long to get results into the mainstream and be reported. So I plead with all of you, that if you're capable of within a driving distance of ALS clinics, you need to participate. I drive 190 miles one way to participate, and I'm in the second study this year.
That's tremendous.
I don't know if everybody else is feeling the same way as I am, but as I listen to the pals that are talking and the doctors and researchers that are talking, I can feel my heart rate going up, and I don't know whether it was partially because I was getting angry or if I just was getting excited about it. I don't understand. We have this gentleman over here that knows that a particular drug is working for him. And we have a gentleman over here that knows that there's a drug that was working for him, whether they were the same, I don't know. But our son has ALS. He was diagnosed a little over a year ago. And he was on a couple of trials, and as it turned out, he was on placebo. And it is very, very depressing and discouraging, especially for the patients, but also for the families. I don't understand what adverse effect could be any worse than nothing. And our families are going, just destroyed. Because of this disease. And when you've got someone who knows that there is something that helps, and that person should be able to make the decision, not the rules and regulations of clinical trials or the FDA or anybody else. They should have the final say. And I totally agree with having trials where the patients knowingly will be given, you know, the drug that is being put up on the trial. Let them make that decision. And also, our son has progressed very fast. And he probably wouldn't qualify now for trials. And I've heard this so many times that people say, you know, as far as their lung function and everything, that, well, they won't take him because he doesn't have the lung function that he did before when he was on the earlier trials. He's the one that needs it as much as anybody else, and he's got absolutely nothing to lose. If he's willing to put himself out there and do whatever it takes to save himself, or if he can't save himself, maybe have the information and the trial and the data to help another person down the road. Well, I spent 30 years in the insurance industry. I worked with human resources. I worked with regulatory departments in every state. The one thing that I learned a long time ago is don't ever say that the reason you aren't doing something is because this is the way it's always been done. We need to change. We need to change things. We need to get things faster. We need to get the FDA to say, let them sign off on their own life. You know, if they want it, let them try it. We have nothing to lose.
I mean, I don't have a good answer for you. I mean, I hear where everybody is coming on this, and nothing that I say is going to answer the emotionality there. And we agonized as a company whether we had to have a placebo, because we didn't want one. But we actually ultimately decided that we had to, because otherwise we would never know whether the drug worked. And we would be 10 years from now, 15 years from now, 20 years from now in the same place. That patients would not know if it works, they won't know if it's safe, and it could actually be worsening outcomes. That has happened. And you need to know that, whether or not it is actually worsening patients' outcomes. And the only way to do that is to do placebo. Now, as we designed it, it wasn't truly placebo. They could take whatever else they wanted to be on for ALS. You could be on Riluzole. And at the completion of our study, everybody got active drug for as long as they wanted. And everybody who has completed the study is now on open drug. So we've done everything possible to minimize the exposure to placebo. But the ultimate goal has to be that we need to define whether it works for an ALS population to start to develop better therapies. And we can't do it without that. And the reasons for that are ALS patients are different from one to another. And ALS treatment has changed over the last 5 years, and it's different. You cannot just compare back to a group and say, this drug works or this drug doesn't work. And if you try that approach, the drug will never get approved and it will not be available to ALS patients.
I don't see any reason why you can't compare the ALS. They are different, exactly as you said. Each one is different, and we don't know why one might live longer than another, or one might lose their voice first, or one might lose their legs first. They're all different, but there's no reason why you cannot compare the data between the ALS patients that are trying the drug. You can still get, you know, the information. You know going in what differences they have. You'll know when they come out of the study what differences they have. And like I said, to worry about an adverse effect, make them get worse, that is the last thing they're thinking about, is that they might not get better or might get worse. Because without it, they're gonna get worse. They're gonna die. And I'm not trying to be, you know, we appreciate everything that you guys are doing. Everything, people all over the world. But you have to leave it, some of it, you have to leave up to the individual to have control over their lives. They just don't have any control over anything else, it seems.
Other questions? Has anyone got a mic on them already? It's hard to see if you, thank you.
I think I'm excited about this whole thing. This is the second time I stood up today. But eventually my question is going to be about the stem cell research. Being a member of one of the largest SOD1 families, this is very virulent in my family. No one has lived beyond a year with it from the time of onset. With the exception of one who was in one of the drug trials. She lived 14 months, which was amazing to me, because no one has ever done it before. The real thing is we know who has the gene and who doesn't in our family. Will this stem cell research showing that it protects the cells ever be used as a trial, at least to start, as an inoculation to prevent its ever showing up?
My short answer is yes. So, as a small company, our resources are limited, and we can only pay for that as one idea at a given time. But barring that, we do have an idea that I think would be suitable for familial SOD1 as the ideal candidates. That is, if we had, we do, that we're working on, it's still in the back burner, and I can't really tell you all about it, but has a cell line that has more potent neuroprotective effect. And we would like to put the cells into the upper motor neurons that has a diffusible factor that can be released and protect the motor neurons across much of the central nervous system. And it is neuroprotection idea, therefore it would be ideal to design a trial where the cells are put in prior to the onset of the symptoms and to see if they can delay the onset of symptoms. And familial SOD1, knowing the genetic diagnosis ahead of time would enable us that such design as opposed to sporadic ALS. Right now it's just only in the idea stage. We need to put together the resources, plan, sites, and put together a hard plan to make that into a reality.
Thank you. Other questions?
Yes, what I would like to see is possibly somebody from the FDA invited here to hear the impassioned pleas from, like this lady talking about her son. The placebo trials, clinical studies, to me, do not make any sense with a disease like ALS. I've been diagnosed with ALS since May of '99. If I was in one of these studies, it would throw off, you know, how would they know with me whether it was working or not? Each individual person is so different, as you've heard everybody say. They're so different. Somebody's voice is affected first, somebody's limbs are weakling. There's no way with a drug trial that you can tell with ALS whether it's working or not. It has to be individualized to each person. Mark here said that, you know, the drug that he was taking was working for him. That's what needs to be, you know, documented. This worked for him. Okay, it may not work for another ALS patient. We don't know, but it did work for him. And the FDA has to know that there's not enough time for individuals with ALS, in most cases, to go through the clinical trials. They have to have the medicine available to them, and they have to have, each individual person needs to be documented what the medicine does for them individually, because none of us are the same.
So I've just looked and I didn't realize how the time has flown. I don't know who has mics, but the lucky ones who have a mic can have the last say in the session.
My question is about Biogen. Do you have an update on when trials for that would start in ALS patients?
Sorry, we missed the first part.
Okay, it's about Biogen, the MS drug, when trials would start for ALS patients.
I don't have a timeline for it. I'm actively involved in designing that trial. We're working as quickly as we can to get that together. There are a number of steps that we have to go through, and because of the sort of known side effects of the drug, they may be a little bit slower to go through each of those steps, but we're actively working on that as quickly as we can.
Would anyone else have
approved for, you know, for MS patients? There's still safety concerns in humans?
So there are known side effects, I guess, and that's better to know about them than to not know about them. And so in that way, we can move a lot faster with a compound like this than we could with a compound that might have side effects that we don't know about. But at the same time, we have to exercise a certain amount of caution, and it just means that there's going to be oversight that we need to respond to as we develop it. It doesn't stop things, but it just means that we need to be cognizant and do all the right things to monitor.
Like maybe this summer those trials might start, or at least, you know, by fall?
I'm sorry, I just don't know the timeline. I just don't know.
So I think with that, I'm sorry to have to close, but I do know that all three speakers will be around for the next couple of minutes. I think the town hall meeting is in here, so things will be transitioning. But I first of all want to thank a tremendous panel, not only for their presentations, but for the commitment they have to this clinical research. Thank you all for your good questions and your participation.
And if we could just have your attention for a few minutes here. We are going to draw, as promised, for the iPad. We have those of you who have been through the exhibit hall, thank you, and have visited with our exhibitors. First of all, I want to thank you. They have had given